So again, this is something that we've introduced, a concept that we've introduced way back in the first part of the semester. So it shouldn't be anything that's new to us, but it's a great opportunity for review. All right. All right, so some quick statements. All living things are made up of what? What's the most basic unit of life? Okay, that's true. All living things are made up of matter. Thank you, Jaden. Everything is made up of matter. But Kyrie is right. This is the answer we're looking for. Cells. All living things are made up of cells. All living things have DNA and RNA to hold blank. What do DNA and RNA store for living things? What kind of information, Nick? Because you're on the right track. You're just missing one word. There's an adjective that should go in front of information.
Genetic information.
Genetic information. Thank you for tag teaming on that, Brian. All living things make proteins through protein synthesis. And this is a process that we will review on the next slide. And all living things complete essential life processes in their own unique way. And those essential life processes, we use the acronym STRNGR to describe. STRNGR stands for those 8 essential life processes that we see universally in all life forms. Although they might accomplish those 8 processes in their own way, we see them still having to do those 8 processes. All right, so protein synthesis was a concept that we covered in our 5th unit just before we took our midterm exam. And we talked about the fact that protein synthesis starts off with DNA, and eventually you end up with a protein. Proteins are what give us our traits. They're what give us our hair color, our skin color. They're even what give us our personality in some ways. So we rely on proteins to make us who we are, but that process starts with our DNA. Along the way, DNA has to first become something else. And that something else is, does anybody remember? DNA has to become something else in this spot right here. Good, excellent, Jaden. It's got to become mRNA. And that M stands for messenger RNA because it's carrying the message from DNA to another part of the cell, from the nucleus to another part of the cell. And that process of going from DNA to mRNA has a name. Does anybody remember what that name is? To go from DNA to mRNA. Excellent, Jessica. That's called transcription. The second step is to go from mRNA to amino acids, and that's called translation, okay? It's important to separate the two. They are 2 different processes. Transcription comes before translation. It's easier to remember because transcription is alphabetically earlier than translation, right? Transcription is earlier in the alphabet than translation would be, or earlier in the dictionary. It's also important to kind of understand what's happening here. So DNA is a nucleic acid, and mRNA is also a nucleic acid. So we're not changing the language here. We're just changing the format in which it's written. It would be kind of like if I were to speak something to you and I asked you to type it out. It's still in English, but instead of being in spoken language, now you're actually writing it down or typing it out on paper. So that's what we mean when we say transcription. Same language, they're both nucleic acids, but they're just in a slightly different format. RNA has U's and DNA has T's. That's what we mean by a different format. And then translation means we're actually putting it into a different language. So instead of it being in the language of nucleic acids, we're putting it into the language of amino acids, all right? And amino acids are different than nucleic acids. We only have 5 nucleotides, A, T, C, G, and U, but we have 20 different amino acids, all right? So it's a slightly different language. That's why we have to translate it. Thank you for letting me know, Jobert. But let me go back. Bottom line is that genes code for the proteins. Genes are the first step. We rely on genes to give us the proteins that we need. They are the instructions. Let's do a couple practice questions. And I want you guys to answer in the chat. The question here is, which most directly affects the sequence of amino acids in a protein? Is it A, the DNA in the nucleus of the cell? B, the amino acids found in the cytoplasm? C, the type of cell in which the protein is made? Or D, the region of the cell in which the protein is made? Which most directly affects the sequence of amino acids in a protein? I've got one answer from Joseph who says A. Fanique also says A. Any other thoughts here? All other thoughts? Well, okay, that's good because Joseph and Fanique are both correct. The answer is A. If we change the DNA, we're changing what's eventually going to be made by the DNA. The DNA codes for the protein. So if I change the code, then I change the protein. I've always liked to think of it in this way, and I'm going to send it to the chat. DNA is the recipe book. Amino acids are the ingredients, and proteins are the meal. So in order for me to end up with the right meal, I need to make sure that I'm using the right ingredients and also that I'm combining them in the correct order. The order matters, right? If I'm making a pizza from scratch, then obviously I don't start off with the tomato sauce. I need to start off by making some dough, and then I can put the tomato sauce on top. All right, so the order also matters, the ingredients matter, but ultimately my instructions, my recipe is coming from DNA. And that means that if I change the instructions, if I change the recipe, if I change the DNA, then ultimately my meal is going to be changed as well. So the answer to this question is DNA. Thank you, Fanique. Thank you, Joseph. One more. How many amino acids are affected by the deletion of one nitrogen base? A, all amino acids are affected. B, no amino acids are affected. C, all amino acids before the point of change are affected. Or D, all amino acids from the point or after the point of change and beyond are affected. what would you say? Which amino acids are affected by the deletion of one nitrogen base? You can answer in the chat. Okay, we've got Nick saying C. We've got Joseph saying D. We've got Jessica saying C. Are there any other thoughts on this matter, on this question? All right, D is the correct answer here. So we're gonna see, I'm gonna demonstrate this on the next slide, but this is called a frame shift mutation. Frame shift mutation. What happens is that when I delete one of the nitrogen bases, everything that comes after that is going to be affected. The reason for it is that we read nitrogen bases, or we read DNA, we read genes in codons. We read them in groups of 3. So if I delete one of those bases, now my group of 3 has to shift over, and we're going to see that take place. It would be like if I had the sentence, the fat cat ate the bat, or the fat cat ate the rat. We read words and we read the codons in groups of 3 like that. The fat cat ate the rat. Now, if I deleted the F in the word fat, I still have to read in groups of 3, but now it looks like this. I deleted the F, right? Look in the chat. I deleted the F. The fat cat ate the rat. I deleted the F, but I still have to read it in groups of 3. So now it looks like the ATC, ATA, tet, her, at. The sentence doesn't make sense anymore. Everything that happened after I deleted the F is now messed up. Everything before I deleted the F is okay still. It's how it should be. Let's say I deleted the T in the word ate. So now I have the fat cat. Now again, everything that happens before the T is where it should be. The fat cat, AET, her, at. So the first 3 words are good, but then at that point, everything's messed up. So let's see what this looks like in actual genetic terms. So up here, I've got a strand of DNA. I know that it's DNA because I have T's here. DNA has T's, whereas RNA has U's. So my strand of DNA says T, A, C, G, G, C, C, G, T. The first thing I need to do is transcribe that into mRNA. T is transcribed to A. A gets transcribed to U because RNA does not have any T's. We can't have T in RNA. So A is transcribed to U. C gets transcribed to G, and G is transcribed to C, and so on and so forth. So I end up with A, U, G, C, C, G, G, C, A. And then I would use my codon chart, or otherwise known as my amino acid chart, to figure out what amino acids I'm dealing with here. So this is something that you might need to review if you're like, I remember doing this, I remember looking at this, but I don't remember how to do it. This is something you should definitely go over because I can almost guarantee it will be on your EOC exam. But AUG, so I start off with my first letter, A, and that tells me that I should be somewhere in this section here. This is the correct amino acid that I'm looking for. My second letter is U, so that tells me that I'm looking in this small section right here. And then my third letter is G, so I follow my G over, and that tells me that I end up with methionine as the amino acid. My next codon is CCG. Remember, codons are read in groups of 3, CCG. My first letter is C. My second letter is also C, so I'm now looking over here somewhere. And my third letter is G, so I end up with proline. And next, my next codon is GCA. My first letter is G. My second letter is C. And then my third letter is A. Alanine is my amino acid in this case. All right, so methionine, proline, and alanine. Now, let's see what happens if I delete the fourth base. I want to get rid of this G. I don't want this G here anymore. I'm deleting that guanine. This is what I end up with. Remember, codons have to be read in groups of 3. So I can't just delete this and just read these two right here. I can't just read this C and this G now. I need three nucleotides to give me an amino acid. So everything gets shifted over. Everything ends up being shifted over. So methionine is still the same. I still had AUG because that happened before the point of deletion. But then after that, instead of having CCG as my codon, I had CGG. So instead of getting proline, I end up with alanine. And then what happens after that? I should have GCA as my next codon, but instead I have CA, something else over here. So I'm thrown off again. Everything after this point of deletion is messed up. So that's what a frame shift mutation is. Everything after the point of deletion is messed up. All right. So Sternger is a great starting point for a study guide. And I would really encourage you all to take a picture of the screen, to take a screenshot of the screen, and to use it as the foundation for your study guide as you prepare for the EOC. Sternger are these 8 life processes that we began talking about way back in August. But we've learned so much since then. We've covered so many topics, and we've introduced so many concepts and vocabulary words and images. But the cool thing is we start off with Sternger and we end off with Sternger as well. And that means that you can use it to try to summarize all those other concepts that we've gone over this year, this semester. If I were you, I would consider taking my notebook and making each letter of Sternger its own page. Synthesis gets its own page. Transport would get its own page, and so on and so forth. And I would try to relate as many concepts from our unit, from our course, to each of those letters. Essentially, you could touch on everything we've talked about for the last 3.5 months from the template of Sternger. All right, so please make sure you have this recorded somewhere. Please also be aware of the fact that our YouTube page is up and available for you all to refer to as a review mechanism. And we're going to continue to review in class as well. Before we play this Kahoot, I want to go over this because this will really help you all with the Kahoot. It'll put you in a good position to do well in the Kahoot. So we've got these 4 biological compounds. This is what we talked about in our very first unit way back at the end of August and in the beginning of September. 4 biological compounds: carbohydrates, lipids, nucleic acids, and proteins. I asked you guys way back then to copy down this chart. And I remember Kyrie definitely had it copied down because he was able to give me the next day almost all of these answers. So what I'm looking for now is for those of you who have been diligent note takers to flip back in your notebook and go way back to early September, late August, find this chart so we can fill it out again before we play this Kahoot, which is basically going to be all about this chart. So I'm giving you about 10 seconds to flip back in your notebook and find this information. All right. So can someone give me this first box? What's the monomer of a carbohydrate? What is the monomer of a carbohydrate? Otherwise known as the subunit or the building block of a carbohydrate. Thank you, Alyssa. Yeah, glucose. Glucose is the monomer of carbohydrates. Now, those of you who maybe joined the class later in the semester, or you weren't present for class for those days, or you lost your notebook, or you can't read your handwriting, whatever it is, if you don't have this information readily available to you, this is now your time to write it down, to take a screenshot, to take pictures. This is foundational information. I guarantee you several pieces of information in this chart will be on your EOC exam. All right, so let's be active learners here, active participants in this lecture. Don't just let Alyssa and Joseph and Jessica and Fanique answer and Brian answer all the questions. Contribute here. Box number 2, what are the elements that make up carbohydrates? What elements make up carbohydrates? Now, there are 3 elements that are present in every biological compound. Can you give me at least one? Excellent, Alyssa. Carbon, hydrogen, and oxygen. Now, the special thing about carbs is that those elements appear in a 1 to 2 to 1 ratio. Thank you, Jessica. They appear in a 1 to 2 to 1 ratio. So what this means is that I have 1 carbon that guarantees that I will have 2 hydrogens and 1 oxygen. If I have 2 carbons, that guarantees that I will have 4 hydrogens and 2 oxygens. If I have 5 carbons, that guarantees that I will have 10 hydrogens and 5 oxygens. That's what we mean by a 1 to 2 to 1 ratio. They remain in sync with one another in that way. What's the function of a carbohydrate? What is it supposed to provide for living organisms? Good, Jessica. And what kind of energy specifically?
It gives provides air. It gives you air to breathe.
Incorrect. That's not true.
Incorrect. All right.
But I appreciate you for speaking up, Anthony. So we're looking for a specific type of energy. Carbs are responsible for providing living organisms with energy. Joseph, did you say something?
I said short-term energy.
Short-term energy, otherwise known as quick energy. Good. So we don't like to store carbohydrates. If our body has to store carbohydrates, it's not typically a good thing. It can lead to fat. And what would be an example of a carbohydrate? What's an example of a carbohydrate?
Carbs and food.
Okay, yeah, that's what carbs are. Carbs are carbohydrates. Glucose is an example. We've got that as the monomer. What else can we think of? What is another example of a carbohydrate?
Sugar. Glucose. Sugar.
Sugar, yeah. So all of these are sugars. Sugars are carbohydrates. And typically, when we see an ending OSE, that's telling us that it is a sugar. Sucrose is a sugar. Fructose is a sugar. Glucose ending in OSE is a sugar. Also, starch and glycogen are carbohydrates as well. All right, next one, lipids. What's the monomer of a lipid? Come on, people. I know we know this stuff. Or we should have it written down. Even if we can't actively recall it, it should be somewhere in our notebooks. Glycerol and fatty acid tails. Thank you so much, Alyssa. Thank you for adding to that, Jessica. Good. The elements are the same, carbon, hydrogen, and oxygen. But the difference is that for lipids, they don't exist in a 1:2:1 ratio. All right. What about the function of lipids? What do they do for living organisms?
They store energy.
They store energy. They provide long-term energy. They're also a vital part of membranes. That's also important to note. That's connected into our second unit when we talked about cells and organelles. Lipids make up the cellular membrane. What would be an example of a lipid? What's an example of a lipid?
Oils, waxes.
Oils and waxes and steroids. Good. All right, jumping into nucleic acids. What's the monomer of a nucleic acid? DNA is an example of nucleic acids, Nick, so keep that in mind for the 12th box. But what would be the monomer of a nucleic acid? What makes up a nucleic acid? Good. Nucleotides. Thank you, Jessica. Now we've got those same three elements, carbon, hydrogen, and oxygen present in nucleic acids, but they also have two other things that are very important. Does anybody remember those other two elements? Two additional elements that make up. Good. Phosphorus is one. Thank you, Fanique. And we're missing one other. Starts with an N.
Nitrogen.
Nitrogen, good. Okay, and what's the function of nucleic acids? What do they do for us? We kind of touched on this earlier. What do nucleic acids do for living organisms? What does DNA do for living organisms? We talked about this earlier in the lesson. They control cellular activities, and then ultimately they control the production of proteins. Good. And then Nick already told us that DNA is an example, but we also have RNA as well. And there are different types of RNA: mRNA, tRNA, rRNA. Lastly, we've got only 4 more. So proteins. What's the monomer of a protein? Should be quick. Excellent, Fanit. Thank you. Amino acids. What are the elements in proteins? We've got those same 3, carbon, hydrogen, and oxygen. And then there's one more.
Glucose. Sugar.
Glucose is not an element.
Nitrogen.
Okay. Nitrogen is the one we're looking for here.
Oh yeah, I knew that.
Anthony, I don't think you were in the class at this point when we went over this. So please do make sure you take a picture or a screenshot of this.
Okay, I'm gonna do that right now. Screenshot. Yeah.
Okay. The function of proteins. What do proteins do for us? They do a lot of things. They're the do-it-all compounds. They help us to fight off pathogens. They help us to speed up reactions. They help us to break down compounds. They help us to build up compounds. Proteins are extremely important. And we've got some basic examples: enzymes, hormones, insulin, and antibodies. All right, so what I want you all to do is, again, take a picture, take a screenshot. I'm going to leave this up for a few seconds while you do that, because this is very important, and then we're going to play the Kahoot, which ties directly into it. right. Let's play this Kahoot. This might take us to almost the end of class, but hopefully you all can use the practice. So you're going to go to www.kahoot.it and enter the game pin through 5397639. www.kahoot.it, game pin 5397639. I'm only going to wait about 60 seconds to allow people time to join this game. Thank you, Kyrie. Thank you, Fanique. Thank you, Alyssa. Thank you, Anthony. Thank you, Nevaeh. Thank you, Joseph. Thank you, Jessica. Thank you, Nick. Thank you, Makaran. Let's keep going. Come on, we got 10 seconds left to join. Thank you, Jobert. All right. Which of the following is not a function of proteins? We just went over this stuff. Which of the following is not a function of proteins? Come on, folks. So you all chose every answer, except for one of you, that was incorrect, that the proteins actually do. So remember, proteins provide a lot of function to living organisms. The only thing that they don't do in this list is store and transmit heredity. That means storing and transmitting genetic information. That's the job of nucleic acids, not proteins. Excellent job, Joseph. Everybody else, let's get going. Enzymes affect the reactions of in living cells by changing the what? How do they affect the reactions? What do enzymes do? Yeah, enzymes change the speed of the reaction. They zoom, they speed things up. Good. Joseph's still in the lead. Which of the following organic compounds is the main energy, the main source of energy for living things? All right. So I'm not sure why we had so many people saying nucleic acids. They do not provide energy. We just went over this. Carbs provide short-term energy. Carbs are really what most forms of life rely on for energy. Glucose, right? Plants and all photosynthetic organisms do photosynthesis to create glucose, which is a carbohydrate. Lipids also provide some energy, but they're really about long-term energy storage. It's not really, they're not as useful as glucose for immediate energy use. Good job, Nick. Big jump. The most abundant element in most living things. Okay. So oxygen was also a good answer because oxygen is present in all four of the biological compounds, but carbon is what we would consider the organic element. It's what we rely on in all forms of life. Good job, Fanique. Keep moving up. Good job, Kyrie and Makarin. A monosaccharide is a what? Okay, good. Monosaccharides are carbohydrates. Okay, good job, Anthony. Keep climbing. A single unit that joins together to form a polymer is called a monomer. So we take several monomers, we put them together, we create a polymer. Seems pretty simple, right? A monomer, mono means one. If I take several monomers and I link them together, I create a polymer, which means many. Come on, folks. Substrates fit into the blank blank of an enzyme. Substrates fit into the blank blank. Okay, excellent. Yeah, we did well on that one. The active site is where substrates fit. Good, that's where the action takes place. Good job, Nick. Keep going. What is the monomer of a carbohydrate called? Just looked at this. What's the monomer of a carbohydrate? Good, a monosaccharide. The monomer that makes up a protein is called a, or an, what? Good. An amino acid is the monomer that makes up a protein. All right, let's keep going. Glycerol and 3 fatty acids make up what? Good, lipids. Wow, Nick, 6 in a row. Keep going, keep going. Let's see. What is the optimal temperature for this enzyme? The optimal temperature. Where is it working the best? Good. So we look at this image again. The optimal temperature is where the curve is the highest, and that appears to be right at 40 degrees Celsius in this case. Nick is on fire. Good job. DNA and RNA. What are they? DNA and RNA. They're both nucleic acids. That's what the NA in DNA and RNA stands for. All right, everybody's jumping up now. Good job. Come on, Kyrie, stay in it. Starches are what? Starches are carbohydrates. They're examples of carbs. Good. Okay, looks like Nick may have lost the streak. Fats and waxes. What are they examples of? Fats and wax, or they're lipids. Good. Oils. What are they an example of? They're also lipids. Good job. Let's keep going. Kyrie's making a comeback. Sugars. What are they an example of? Sugars are also carbs. Jessica's making a comeback as well. Joseph is still in a healthy lead. Biomolecules contain atoms of, there's only one real answer here. Good, carbon. Fanique, 12 in a row. Wow. Good job. Biomolecules exist in what? Yeah. They're present in all living things. Now, you could also make a case that you might find some in the rocks and in the dirt as well. But we want to think about life. That's why we have this root word bio present. Enzymes blank the activation energy of a chemical reaction. What do they do to the activation energy? Okay, that one got a few of you. So, well, we didn't have an image, but enzymes speed reactions up by lowering the activation energy. It would be like lowering a hill that you're trying to run up. If you lower it, it's not going to take you as long, nor will it take you as much energy to get there. So you've got to lower that hill. That's what enzymes do. Oh, Fanique got it right. Keep going. Okay, she's catching up, Joseph. Blank are catalysts. What are catalysts? Good. Enzymes are the catalysts. A catalyst is something that speeds things up. McCarran's got a streak of 4 going as well. Enzymes are very specific and only work with certain blank. That should be work with. Okay. Yeah, they only work with certain substrates. Keep going. Okay, everybody's got some good scores going here. Good job. Enzymes blank blank chemical reactions. They speed up chemical reactions. Good. That was kind of a give me. Good job. 7 more questions. A substance that increases the rate of chemical reactions without any permanent change. We're kind of stuck on the same topic here, but all right. The catalyst speeds it up without any permanent damage. What happened, Fanique? Okay, come on, keep going. 6 more. Is the substrate blue or white here? The substrate. Okay, that was a tough one. Now, you could probably make a case that either one could be the enzyme, but typically the substrate is going to fit into the enzyme's active site. So the enzyme in this case is white and the substrate is blue. Let's keep that in mind for this next question, which asks, is the enzyme blue or white? All right, good job. Yep, the enzyme was white. Wow, Joseph's going to crack 20,000 points. Good job. All right, what is this a picture of? Which of the 4 macromolecules is that a picture of?
I can't really see it.
You should just know that it has a backbone and tails. That's all you really need to be able to see. A backbone and tails. All right. Yeah, that was a tough one. That was a lipid. That was a picture of a lipid with the glycerol backbone and the 3 fatty acid tails. Good job, Joseph. 3 more questions. What is this a picture of? Yes, that is a carbohydrate. That's a picture of glucose, actually. All right, let's see if Fanique can crack 20,000 as well. We've got 2 questions left. What is your favorite holiday? Any answer is correct. Just go as quickly as possible. Good, everything was correct there. Everybody says Christmas. I guess maybe we're just thinking about Christmas appearing, or maybe it is actually everybody's favorite holiday. We'll see. All right, Fanique, I don't know if you're going to get to 20,000 unless you answer this one right away. At what pH will lipase be denatured at? All right, let's see the final tally here. See this podium. Good job, Nick. 19 out of 29. That's pretty good. 23 out of 29 is good job. Is a good job too, Fanique. And then 26 out of 29, Joseph. You killed it today. Excellent job. That's basically the end of our class period today. There is an asynchronous assignment for you all, so don't forget to give that a look. It's actually pretty easy. Shouldn't take too much time at all. As I said, I'm done updating grades in Canvas. Now I just have to take them and put them into PowerTool. The assignment for today is called U10D1 EOC Prep Assignment 1. All right, so give that a look over the weekend. It's pretty quick. I appreciate your participation today for the 10 of you who played the game. And everybody have a good weekend. Let me know.